reason, the duck-billed platypus emits a shrill that some people say sounds a lot like Justin Bieber. You don't have to know that, okay? Now, seriously, what I'm going to do is I'm going to ask you basic concept, overall concept with some details, okay? For example, I'm not going to ask you the difference in molecular weight between IgG and IgA, but you should know the difference functionally between IgG and IgA, okay? I'm not going to ask you the enzymes that render a blood type A or B, okay? But you should know what the difference is between what makes red blood cells of type A different than type B, okay? I mean, I know I'm not telling you exactly what's on the exam, but, I was told that I'm not supposed to do that, so. All right, so what I'm going to cover today is, you can argue that this is not really the concept of basic immunology, but the immune system plays a major role in these particular issues. And that is, once again, the immune system doesn't know what's bad or good for you, okay? It only knows what belongs and doesn't belong. And Mother Nature had never designed the transfer of a large number of cells or a whole organ or a whole tissue from one individual to another. In other words, Mother Nature tries to really prevent this. Now, humans, on the other hand, have exploited this concept of transferring something from one individual to another in order to achieve some sort of medical benefit, okay? Replacing a failing organ or replacing blood loss. So when we talk about transplantation, we talk both about blood transfusion and also organ transplantation. I'm going to cover the immunology of such procedures. So what are blood groups? Basically, blood groups are defined by a bunch of different antigens on your red blood cells, okay? And there's a whole host of them that can be defined as unique to a particular individual. Now, fortunately, the ones that are clinically relevant, the ones that make a difference in transfusion, are limited in number, okay? Now, that's not the case when we talk about organ transplant, as we will see in the second part of the lecture. So the antigens that are relevant clinically, okay, are characterized basically by different entities, sugar moieties, on the surface of red blood cells. And this defines what we call the ABO group. So basically, the ABO system, the differences between these blood groups, sometimes they're called blood types, that's an older term, are defined by either the presence, are they there or are they not, and the nature, what kind is there, of particular carbohydrate moieties that are attached to the end of a stand chain of a particular red blood cells component, okay? So depending on what carbohydrate moiety is attached to this particular end stem chain of a red blood cell component determines what kind of blood group you belong. So, for example, the A antigen, and by the way, this is one of those cases when you don't have to remember the enzymes, okay, or the sugars. The A antigen has a transferase that adds a particular N-acetylglycylglycosamine to the end chain. So if you have this particular sugar, you're defined as group type A. Okay, type B, on the other hand, has a different sugar at the end of the particular stem chain, and that's defined as type B. And if you have neither, then you're defined as a blood group O. So here is a simple picture of where you have the end of a stem chain that is, belonging to a particular component. Don't worry about what it is, okay? So basically, if you happen to have N-acetylgalactosamine, you're type A. If you happen to have galactose, you're type B. If you have neither of these, you're type O. Okay, simple as that. So if you have, the way you should remember, forget galactose, is if you have type A, you're A. If you have type B, you're B. If you have neither, you're type O. And of course, if you have both, then you are blood group AB. Simple as that. Now, these are the genotypes that can generate a particular type or a particular blood group. okay? And these are the different genotypes can generate A. So for instance, you could have a mother and a father that are A and B and generate O because they have this expresses a particular genotype right here. The other clinical relevant antigens are those that belong to the RH group. They used to be called rhesus group. It's a very old term. Okay? And there's a whole bunch of different ones of those too, but the most clinical one is the one that I discussed when we talked about the hemolytic disease of the newborn. Okay? So there's a whole bunch of different antigens, but 5 major ones, 5 that are clinically relevant, are labeled as capital or small letters D, C, C, E, or E. Okay? Now, the capital letter D, the antigen that is described by convention as the capital letter D, is the one that is most antigenic, meaning that if transferred to another individual that recognizes it as foreign, is the one that causes the most trouble. And we talked about, once again, the hemolytic disease of the newborn when you have an Rh negative mother with an Rh positive fetus. So the positivity of Rh, whether you are Rh positive or Rh negative by convention, is defined as either you have the capital D antigen or you do not have the capital D antigen. So whatever the composition of those 5 components that I just showed you, if you have the capital D antigen, you're Rh positive. If you don't have it, you're Rh negative. Now, sometimes you'll find this in some books or some chapters, okay, that the absence of a capital D antigen is described by losing a lowercase d. Okay? Again, I didn't make these rules. Okay, that's the way it is. So in other words, the little d is used to indicate the absence of a capital D. So if you see these denominations, okay, remember this pattern. Okay? If there is a capital D in the composition of the Rh antigens, once again, you're Rh positive. If there is no capital D, or if there happens to be a lowercase d, you're Rh negative. Okay? So for example, I happen to be B positive, which means that I have the B antigen on the surface of my red blood cells, and I also happen to have the capital D antigen on the surface of the red blood cells. Now, there's a whole host of other erythrocyte antigens that are defined by various different categories that also have some clinical relevance, but at this point, you know, this is well beyond the scope of this lecture to cover them all. Now, what is the significance about this? The significance is blood transfusions. Once again, Mother Nature never designed to have a large amount of red blood cells transfer from one individual to another. Okay? Humans have created this concept in order to replace a large loss of a large number of red blood cells. Now, what is the problem is that some of these antigens, okay, that make the blood groups, the ABO group, are also common antigens found anywhere in various different other things in nature, okay? So you have antibodies to these antigens as an individual because you have been exposed to these antigens on other sources, for example, some intestinal bacteria. Now, the issue is, of course, you're not going to make antibodies to your own antigens because of the process of immunological tolerance. Remember that? So you have naturally occurring antibodies to these blood antigens except for yours. Once again, why? Well, because the immune system tries to eliminate cells that are self-reactive. Okay? The problem comes when you are given these antigens by having transfusion from another individual. Okay? You do have circulating antibody against somebody else's blood group. So these are the groups and their serum antibodies. So, of course, if you are group A, that means you have antigen A on the surface of your red blood cells, okay? You're not going to have antibody to A because it's your own, but you do have antibodies to B. Vice versa, if you are group B, that means you have a B antigen on the surface of your red blood cells, so you're not going to make antibodies to your own, but you do make it against A, okay? Now, if you are AB, then you have neither of these antibodies circulating because you have both an A. On the other hand, if you're type O, because you have neither A nor B, you have circulating antibody against both A and B. I want you to remember this pattern. Okay? So put an asterisk next to this. Now, what does this mean? What do you do when you give a blood transfusion? What is the scope of a blood transfusion? Okay? It's not to really replace neutrophils, okay? It's not really to replace macrophages. It's to replace a large number of red blood cells that you may have lost for one reason or another. For example, some physical trauma. So the scope of a blood transfusion is to give you, the recipient, a large amount of red blood cells to replace the one you have lost. So the most important thing in a blood transfusion to be aware of is that the recipient does not react against the donor cells, okay? Or, saying in a different way, the donor cell must not react with antibodies in the recipient's serum, okay? You don't worry about the antibodies present in the donors, okay, because there's not a lot of them. But in the recipient, you do not want the donor cells to be attacked by the recipient serum. Remember that, okay? So you don't want the recipient reacting against the donor red blood cells. The reason is, okay, and this is, by the way, the pattern of who can give to whom and who can receive from whom. And I want you to know this pattern, okay? So if you are group A, okay, because, okay, you can give to A and AB and you can receive from A and O. Why? Because A is not going to have antibodies to your own because it's the same as your own, okay? And also, O doesn't have anything on it, okay? So it doesn't have antibodies to either A or B, okay? Same thing if you're group B, okay? You can B can receive from B because it's the same, and O can receive from B because O doesn't have an antibody to either A or B. Do you see the pattern? Once again, you gotta make sure that the recipient serum does not have antibodies against the donor cells. Okay, so this is the pattern. Okay? So obviously, O is the universal donor. Why? Because excuse me, because it doesn't have anything on its surface, it's not going to react with any antibodies. But on the other hand, it can only receive from O, okay, for the same reason, because O will not react to its own self. Okay? So make sure that you understand this pattern. Now, why do you want to avoid a reaction of the recipient against the donor red blood cells? Okay? Because what happens is if you have a very strong reaction of the recipient against the donor red blood cells, you have a massive immune reaction to a large number of antigen-looking red blood cells, okay? And this in turn causes, in many cases, immediate hemolysis of the red blood cells. Now, when you have a lot of broken or hemolyzed red blood cells, that initiates a cascade of events that in some cases can actually even be fatal, okay? So, for example, these are caused by antigen-antibody complexes on the erythrocyte membrane. Remember, you have antibodies in the serum of the recipient reacting to the donor red blood cells. And these, in turn, causes a cascade of events. No, I'm not going to ask you to remember Hageman factor, factor 12A. Just know that in some cases, the immune reaction of the recipient against the donor red blood cells can cause a lot of different problems, okay? And these are just one of the many cascades that can occur in the most severe cases when you have a mismatch. Okay, and here's some more. Now, how do you know then what type of blood group you're a part of? Well, it's very simple, okay? It's usually done in the lab, and what happens, that either antibodies bound to the erythrocytes, okay, or more importantly, the antigen of the red blood cells can be detected by very simple laboratory procedures. Okay, so what happens is you have type screening and cross-matching. Type screening simply means taking a donor red blood cells, okay, a small sample of red blood cells, and mixing it with commercially available antibodies against a particular blood groups, okay? So you can purchase antibodies that are anti-A, anti-B, or anti-D, okay? And what happens is that if you mix these commercially available antibodies, this used to be done on a microscope slide. Now it's done in little test tubes, gelatinous test tubes. I'll show you that in a minute, okay? So if your red blood cells clump up when you mix it with anti-A, then simply you are A. If they clump up when they mix it with anti-B, okay, then you are B. If they clump up with both, you're AB. If they don't clump up at all, and then you are O. And this is the same thing with a D antigen. You have an anti-D antibody, commercially available preparation. If they clump up, then they are positive for D. Simple as that. Okay, and here is a picture of, this is a gelatinous material. Okay, if the cells clump up, they don't go through the material upon centrifugation. If they don't clump up, then they fall to the bottom, okay? And this is usually done to determine blood type, okay? So this is AB negative. These are the controls. See, it comes and clumps up with A, it clumps up with B, but it doesn't clump up with anti-D, okay? So this is an antibody against A, this is an antibody against B, this is an antibody against D, and these are the controls, okay? So here it's clumping up with A, it's clumping up with B, but it's not clumping up with D. This individual is AB negative. Simple as that. Okay? This is how usually you determine blood type. All right, so what about organ transplantation? Well, organ transplantation is much more complicated for various different reasons. Okay? Because once again, you're transferring something, a complex entity from one individual to another. Now, this has a fairly recent history. You're not talking about hundreds of years. Okay? So the first trial of organ transplant, I think it was kidneys, I don't remember exactly, but it was attempted in the 30s, and of course the organ from one individual to another was immediately rejected. There was an immune reaction to the organ didn't take. Then the first successful kidney transplant occurred between identical twins in the 50s, which suggested that obviously there was a genetic basis of accepting a particular organ, okay? They were featured from an individual that were accepted in another individual simply because those two individuals were identical. And of course now it's fairly routine, okay? There's a whole bunch, I don't know what the numbers are, this is not important, but obviously, you know, the organ transplant is almost an everyday practice in modern medicine. Now, the barriers of organ transplantation is one, the immune system, and we'll talk about that, and also the lack of availability of organs, okay? I mean, you can't just get a human kidney and go on a tops across the street and get it, okay? So it's a limiting factor is how many donors there are out there. Okay, now these are just some of the organs that are routinely transplanted, okay? Just to show you, you know, they're quite common. Some are easier than others. Cornea transplant, for instance, do not require immunosuppressions. Most other ones do. Now, when you transplant an organ, okay, the transplanted organ is called a graft. And there are different types of grafts, okay? An autograft is basically a graft that is shared within a single individual, okay? For example, in burn patients, many times they transfer a piece of skin from one part of your body to cover the burned skin on the other part of your body. Now, obviously that's accepted because it's within the same genetic individual. Then an isograft is from a genetically identical individual, such as a identical twin. Most grafts are called allografts. In other words, they are taken from one individual and placed into a genetically different individual. And that's the bulk of the graft that occurs nowadays. Now, there's also much research in xenografts, which are grafts from the same species, from different species, excuse me, from pig to humans, from non-human primates to humans. Okay, and we'll talk about those. Now, once again, because the immune system works the same way regardless of the antigen, okay, usually you have a biphasic response to a transferred graft. Okay, so you have a first set rejection, which is the first time you see a particular graft, and then you have a second set rejection if a second graft from the same individual is transferred into the same recipient. Okay, so here is a picture, a cartoon of what happens. Okay, so you have, let's say this is happens to be skin. Okay, this is an autograft, meaning that it is a graft that is accepted because it's within the same individual. So you basically replace the piece of skin. There is revascularization. Blood vessels now arrive and revascularize the area. There's some sort of healing, and then you have resolution in which you have complete acceptance of the graft. However, in a first set rejection of an allograft, you have a grafted skin, you have revascularization, but now this is seen as foreign, so you have an infiltration of all kinds of different immune cells, okay? This in turn kind of cuts off the particular revascularization and also damages the graft, and the graft is now rejected. Now, this happens the same way in a second graft rejection, it's just that it happens much faster, okay? Just like a secondary immune response. Now, what is it that causes a graft rejection? In general, it's mediated by cells, not antibodies. Now, this is not necessarily true in a sense that antibodies do play a role, but the major players on the rejection of a graft are cells, and particularly T cells. Okay? In addition, both CD4 and CD8 positive T cells are involved in the rejection of a graft. Okay? Each contributes some to the rejection, but in general, most strong rejections are mediated by a combination of both. Okay? Now, the issue here is, what is it that makes a graft foreign looking? Okay? The problem with blood groups is that there's a limited number that are clinically relevant, the ABO and the RH. Okay? And those are hard to define because there's a limited numbers of entities that you have to watch for. On the other hand, when you're talking about a graft such as an organ, skin, kidney, lungs, etc., the histocompatibility, the compatibility between two individuals are based on their MHC composition, their composition of their major histocompatibility complex. Okay? And that becomes much, much more complex. In other words, in a typical outbred family, outbred meaning not cousin to cousin, marriage, when they're completely genetically different, even the kids have no more than 50% chance that are compatible with each parent, okay? And only a quarter of these kids will inherit identical MHC haplotypes, okay? Now, why is this a particular problem? Okay, here is the human HLA complex, or the MHC, okay? And no, I'm not going to ask you to remember these names. Just know that there's a whole bunch of different components divided into 3 different classes. And here are the combination of possible haplotypes within those particular genetic makeup between 2 parents, okay? So each has their own haplotype, okay? Made in yellow, pink, blue, and red, or whatever this color is. Now look at the progeny, okay? There's all kinds of different combinations that they could get. Okay, for example, this little kid right here got the yellow from the father, and they got this bluish thing from the mother. On the other hand, this kid right here got the yellow from the father and the red from the mother, okay? So the take-home message of this particular slide is that the issue of the genetics of transfer MHC is much more complicated than blood groups. And sometimes you even have recombination. So if you look at the possibilities, again, don't worry about these names. I'm not going to ask you to memorize this. Just know that now you're looking at the yellow, the pink, the blue, and the recombinant. Each of those has their own alleles. Okay? So now you see the complexity. Once again, in the ABO system, you got to worry about two antigens, A and B, and that's it. On the other hand, in this particular system, when you're transferring an organ, you got to worry about matching all these, okay, among different individuals. And that's very complex. And this is why, by the way, in most cases, in organ transplant, you need some immunosuppression because you're always going to have some sort of immune reaction to a graft. So how do you know where two individuals are histocompatible? In other words, how a donor and a recipient are compatible enough immunologically so that the donor can give the recipient a graft, okay? Well, we talked about ABO typing, and now what we do is we go HLA typing, okay, and the mixed lymphocyte reaction, which is an older method of obtaining information about histocompatibility. So tissue typing simply involves the following, okay? Since we are concerned, unlike the blood group, which only has to worry about 2 or 3 antigens, since we're worried about the antigens on the surface of the cells of a graft, okay, that are numerous and diverse, one way to do it is to ask the questions, what are these antigens? What are the proteins on the surface of the particular cells of a particular graft that may be not compatible? Or you can ask, what is the composition of the genes that encode for these proteins? Okay, so in this particular case, you're asking, what HLA or MHC components does this individual have? In this particular thing, you ask, what are the genes? Okay, it's asking the same question, basically. And what you do, okay, is an older version of this was called the microcytotoxicity test, okay? So what you did, okay, in this case, is to ask the question, on the surface of the cell, let's say of the donor, what are the HLA proteins? So what you did is you took white blood cells from a donor or from a recipient, and just like you do with blood typing, okay, you added antibodies to particular MHC components. And then you saw whether those antibodies had the ability to lyse the cells. Okay? In blood groups, you agglutinate them. Here, you lyse them. So what happens is you take white blood cells from, let's say, a donor, and you add commercially available antibody to all these little specific HLA and see which antibodies kills the cell. And how do you know that? Because if you add an exclusion dye, just like trypan blue or others, the dead cells take up the dye. Now, so here is an imaginary donor, a recipient, against HLA allele 2 or HLA allele 1. Doesn't matter which one it is. You add antibodies against these two, okay? And then you add the antibodies, you complement, and all of a sudden, this cell right here becomes blue and is killed. That means that it was killed by antibody against allele 2, therefore it must be allele 2. Okay, simple as that. Now the problem with that is that because this system is very, very complex, there's a lot of different HLA proteins, you really don't have every single commercially available antibody to detect every single HLA component. So what you do, okay, oh, and this is just a picture of the same thing, okay? Here is an imaginary set of HLA antigens, okay, imaginary 1, 2, 3, 4, 5, 6, 7, and then these is the recipient, this is donor 1, and this is donor 2, and obviously donor 1 is the same as recipient 1, so this would be a good donor because it matches the HLA composition using antibodies. Now, what you do, and again, don't let me be scared by the slides, get the concept, instead of asking what proteins are on the surface of the cell by using antibodies, because you can't make enough diverse antibodies, okay, against particular HLA types to test them all, now you look at the genome. Okay, now that we can easily sequence DNA and know what the sequence of the DNA is for that particular HLA type, we simply ask the question, what is the composition of this HLA genes on this individual, simply by sequencing, okay? And there's a couple of different methods. Again, don't worry about these names, okay? Either sequence-specific primers, or sequence-specific oligotype probes. And basically, where you're asking the question is, I have an individual, I want to know what his HLA repertoire is. I know the sequence of the DNA that encodes for each of those HLA. I simply sequence the DNA that encodes for the HLA and see what its composition is, okay? And this is done very, very routinely, okay? These are typical gels that are used in laboratories to genotype for transplants, okay? Just to show you an idea of the result of these two different tests would look, okay? So once again, you either ask what proteins are on the surface of this person's cells to give me an idea of what his HLA repertoire is. You do this by antibodies. Or you ask the questions, can I look at the genome? I know the sequence of all these different HLA alleles, okay? Just by sequencing, I can tell what is the composition of this individual. And then what you try to do is you go as closely as you can between a donor and a recipient. And by the way, there's a whole bunch of different data banks, okay? They store a particular person that needs a transplant with his or her HLA features, HLA repertoire. So when the potential donor comes in, they know what to look for, okay? How closely, it's never going to be identical, okay? But how closely can I match the donor and recipient so the donor's organ has a chance of surviving in the recipient? Now, another way to do this is because an immunological reaction is the action of cells against other cells. One way, and an older way to do it, it's still used to some labs, but it's not more common anymore, is to take white blood cells from the recipient and white blood cells from a donor, mix them together, and see if they fight. That's exactly what you do. See if they fight. Okay, you want to know if, you know, two different group of people don't get along, lock them all together in a room. That's basically what it is. Okay, and here if they're gutting each other. Okay, so the mixed lymphocyte reaction is really taking cells from a donor, white blood cells from a donor, white blood cells from a recipient, putting it together, and see if they react with each other. And how do you know they react to each other? Because what you do is you add radioactive thymidine, okay, which in turn is a reflection of the uptake of this thymidine is a reflection of DNA synthesis, okay? And when two cells are fighting, there's a lot of DNA synthesis and lymphoproliferation going on, and this is an indication that they are stimulated each other. Okay? So here is basically two different cells, okay, where you have two different cells put together. And what they do is they proliferate. And this proliferation means that these two groups of cells don't get along. Okay, they are reacting to each other, and therefore this is not a good match. Now this is one way of actually physically testing, okay, some sort of reaction, actual real reaction between a donor and a recipient. Okay? Okay, the problem is that it takes these tests take several days to complete, and sometimes you don't have several days to transplant an organ. Okay, for instance, if a donor is a cadaver, okay, you have to transfer that organ as soon as possible. You can't wait several days for an MLR. So genotyping right now is the gold standard for transplantation. Now, as far as how closely related you need to get the donor and the recipient, once again, you'll never have a perfect match, okay, unless you're identical twins. So depending on which of the HLA components are different makes a difference of the reaction between a donor and a recipient. Okay, for example, class 2 HLAs are generally more important than class 1, even though class 1 are also important. Okay, and of course, sometimes if you have things in an immunologically privileged site, then you don't even need to match a donor and recipient. So here is a graph of graft survival depending on the mismatches of different HLA components, okay? So if you have, here is no mismatch at all, okay? Either in class 1 or class 2 of HLA. And you can see the organ survives, the graft survives pretty well. Okay, now if you start mismatching 1 or 2 components of class 1, you get a little bit of the decrease, but not as much, okay? Now if you have more than 1 or 2 mismatches in class 1, now all of a sudden you're seeing a decline in graft survival. But we'll look at what happens when you have mismatches in class 2. Even just 1 or 2, okay, now you have a drop in survival. And if you have mismatches in all of them, now the graft survives very little. Okay? So the bottom line of this particular slide and the previous one is when you're trying to match histocompatibility with HLA components, both class 1 and class 2 are important. Class 2 happens to be more important than class 1. Now, what happens during the rejection? Well, obviously there's an immune response against the graft, which in turn does what? It rejects the graft. Okay? So usually you have a whole bunch of different immunological entities that attack the graft and try to get rid of it, because the graft, once again, it's a perceived antigen. It's something that is foreign, that doesn't belong there. Okay? So you could have a hyperacute rejection. I'm not going to spend too much time on this. Acute rejection, and sometimes, in fact, many times, you have chronic rejection, which can occur even with immunosuppression after months and sometimes years. This is why sometimes organ transplants have a limited number of years to which they can survive in the recipient. Okay? And here is just a picture of hyperacute rejection of a kidney graft. Okay, where you have a whole bunch of different antibodies and cells, sees this guy as foreign, attacks the particular vascularization of this organ, and eventually you have, trying to block this particular vessel from feeding this organ, and then the organ dies off. Now, you also have chronic rejection, and chronic rejection is long-term exposure to this foreign graft, and unfortunately that really hasn't changed that much throughout the years. Okay? Eventually your body, in many cases of transplant, will reject the organ. Okay? It could be months, it could be years, but eventually in many cases it does happen. So matching is very crucial. Okay? The closer two people are histocompatible as far as HLA, the better the chance of survival of the graft within the recipient. Okay? Now, just like in any other immune reaction, there is a sensitization stage. Okay? There are antigen-presenting cells. There's feedback mechanism between T cells and antigen-presenting cells. There's B cells, macrophages, neutrophils, all the cells we talked about in different lectures that also are in the sensitization stage. And then you have an effector stage in which the immune weapons that I talked about now attack the graft. They could be cell-mediated, delayed type hypersensitivity, cytotoxic T cell-mediated cytotoxicity. Now you also have antibodies and complement. You have a release of a lot of different inflammatory cytokines. All these are classical effector stages, the immune system, functions that the immune system uses against the graft, as if it would in any other foreign antigen. Okay, and here's just a picture of the same thing, okay? Again, I'm not going to spend too much time on this, but here's a graph, and here are all the possible immune reactions that could result in the rejection of the graft because it is seen as a foreign threat. Now, what do you do about this? Well, the standard approach for transplantation in most cases, okay, except for a few ones, is immunosuppressive therapy. If the particular graft rejection is mediated by your immune system, then you want to try to turn down the immune system so the graft has a chance to survive. Okay? And there's a whole bunch of different approaches. Actually, methotrexate is not a mitotic inhibitor, it's a DNA inhibitor. I apologize for the error here. Corticosteroids, T-cell inhibitor. Why T-cell inhibitors? Well, because T-cells are the major regulator immune response, such as cyclosporin and others, okay? All of these require some sort of immunosuppressive therapy. Now, the problem is, remember that when you immunosuppress somebody, you don't immunosuppress the person only against the graft, okay? All these are general immunosuppression. So one of the things you have to watch is to make sure that, you know, the person is not subjected to infections, okay, is protected for infection, because the immune system is also the one that takes care of an offending microorganism. Now, one of the most common forms of transplantation of graft transplantation is bone marrow transplant. Why bone marrow transplant? Because bone marrow is a source of pretty much every blood cell, okay? It has pluripotent and renewable stem cells that can be directed to make a lot of different blood cells, okay? So stem cells from the bone marrow can make neutrophils, can make basophils, can make T cells, can make B cells, can make macrophages. So the reason why you transplant bone marrow is because you're trying to basically almost replace somebody's immune system. Okay, and these are examples of why you would want to do that. Now, this creates a problem, okay, and this is just bone marrow transplant, usually taken from living donor, okay. You immunosuppress the patient, okay, and then you hope that this new bone marrow takes. Now, there is a disease that is very unique to bone marrow transplant, okay, and this is called graft versus host disease, okay. Usually when you're transferring a graft, for instance, a kidney or a lung or you know, some other organ, skin, you are concerned about the recipient reacting against the graft. Okay? The recipient is getting something that looks forward to him or her and try to reject it. Now, unfortunately, when you're transferring bone marrow, what are you doing? You're almost transferring a mini immune system onto a recipient. Okay? So because the bone marrow, you can think of the bone marrow as a mini immune system, not only you have to worry about the recipient reacting to the host, but now you have to worry about the donor cells reacting to the recipient. Once again, because the donor cells, being almost a mini immune system, sees the recipient as foreign, okay, and can mount an immune response against the recipient. And this is called graft versus host disease. Okay? In other words, you are transferring, in this case, an immunologically competent graft, bone marrow, into a recipient. So while the recipient may see the donor as foreign, the donor sees the recipient as foreign too and reacts to it. Okay? And this is a serious disease that has to be accounted for as a potential possibility when you're transferring bone marrow. Now, animals are now being also looked upon as potential donors, okay? Why people are looking at xenotransplantation? Because there's, of course, a low human organ supply, okay? People have to wait a long, long time for a suitable donor. Sometimes they find none and they die, okay? Now, the ones that you want to look at are animals that could make sense in many different ways. okay? So for example, you wouldn't look at a mouse as a transfer because, you know, the organs are simply not the same size, okay? So you look at animals that may be similar to some extent in function and size to humans. Obviously, the obvious one is non-human primates, okay? Pigs are very much now looked upon to see if there is any possibility of xenotransplantation, okay? But of course, this also has all kinds of different problems, okay? But people are indeed looking at as a possibility. Now, some sites do not need immunosuppression because, once again, they are located in immunologically privileged sites, okay? And these are things like cornea, the anterior chamber of the eyes, testis, uterus, brain. We haven't gotten to a brain transplant yet. I'm sure you know people who need one, okay? But anyway, these are immunologically privileged sites. And here is a graft survival rates, okay? I think these are survival rates for 5 years. There's no immunosuppression needed for cornea, but all the other ones are really pretty much need some sort of immunosuppression. Okay. I know how you feel. So good luck on the exam. And we'll see you on Monday.
The corneas being sequestered, would they have higher chances of